The plan of the institution, the budget, the rules for its administration were not calculated merely to pursue cures for the mentally ill. Science itself was also to be advanced. Report of a Rhine Asylum Committee, 1830. Heredity has an undeniably great importance for mental illness and psychical deficiencies, so it is no accident that attention was focused earlier and more intensely on the inheritance question in psychiatry than in any other area of medicine. Wilhelm Schallmeyer, 1918. Genetics has been supported by compelling images. We think first of DNA, whose helical structure, announced in 1953, is still often exalted as the secret of life. For half a century before that, the science of heredity was identified with neat diagrams of green and yellow, or smooth and wrinkled peas, bred by Gregor Mendel in the garden of an Augustinian monastery. The mutant eyes or wings of the fruit fly also assumed an iconic form. Images like these distract us from a science of mass reproduction. The agricultural breeding factories that already had sprung up before anyone cared about Mendel and the industrialized laboratories of recombinant DNA have never been appealing in the way of a ladder swirling heavenward. Graceful curves can only be part of the story. Let the reader cast an eye over the great filing cabinets of data from armies, prisons, immigration offices, census bureaus, and insurance offices that have been brought to bear on the topic of human heredity. Already, by 1830, the investigation of heredity was saturated with numbers. A century later, the data of human heredity still were produced principally in two related institutions: insane asylums and special schools for children who were called feeble-minded. DNA does not flow gracefully in unbounded space, but is bent and twisted to fit onto stubby chromosomes. The science of human heredity arose first amid the moans, stench, and unruly despair of mostly hidden places where data were recorded, combined, and grouped into tables and graphs. In practice, human genetics has always depended on mundane tools to classify and record bodily traits. Phenotypic heredity, which deals in quantities such as egg or milk production, IQ scores, and medical conditions, persists alongside the analysis of genetic factors that may be supposed to code for such traits. Its importance for breeding and other practical endeavors was and remains much greater than is commonly realized. Statistical techniques, from ordered lists and correlation tables to regressions and cluster analysis, have been fundamental to both sorts of heredity research, genotypic and phenotypic. The public knows little of this. A bitter debate in the early 20th century between biometricians and Mendelians about how best to study biological inheritance seemed to end in a victory for genetics, defined by a focus on discrete nuggets of hereditary causation, for which Wilhelm Johansen in 1909 coined the term gene. The new genetics emphasized microscopy, agricultural breeding, and model organisms. Despite geneticists' intense engagement with eugenics and medicine, Homo sapiens was not their preferred organism. It was too resistant to laboratory manipulation and had too long a generation time in comparison to fruit flies, nematodes, and viruses. Historians of genetics, until recently, almost always echoed laboratory scientists and breeders in their focus on genes and then DNA. This book brings historical focus to that other science of heredity, the tradition of amassing, ordering, and depicting data of biological inheritance, especially in humans. The deployment of heredity data in medical and social institutions preceded academic genetics by about a century and continued thereafter as a set of tools and approaches loosely interwoven with classical genetic methods and understandings. In the dance of influence and appropriation, data work was never a passive partner, and in recent decades, It has reclaimed the limelight